are in listen-only mode.
Good day to everyone joining us, and welcome to today's Excellience webinar, Scale-Up Considerations from Formulation Development to Commercialization. My name is Donna Papacosta, and I'll be your host for today. This seminar will run for about 45 minutes, including a Q&A session toward the end. This webinar is designed to be interactive, so please feel free to type in questions and comments throughout the presentation by using the questions function on the panel on the right-hand side of your screen. If we cannot answer your question during the allotted time period, we will send you a direct reply after the live webinar. At this time, all participants are in listen-only mode. The event will be recorded and made available for future download from the Scientific Seminar page on the Excellience website. The presentation slides will advance automatically. If you need technical assistance, please contact GoToWebinar at the numbers displayed. In the U.S., that's 800-263-6317 or 1-805-617-7000. Now we'd like to begin the formal presentation. Excellience is a premier provider of formulation development and manufacturing solutions with a solid reputation for accelerating early-phase small molecule development. Our formulation development scientists have considerable experience overcoming challenges associated with physical and chemical properties of drug substance or limited quantities of active pharmaceutical ingredient, API, in a manner that results in compounds with improved solubility and bioavailability. Excellience operates out of a CGMP-compliant, FDA-audited, DEA-licensed facility in Tampa, Florida. For more information, please visit the website at www.excellience.com. I'd now like to welcome our speaker for today's seminar, Dr. Paul Skultety. Dr. Skultety is Director, Pharmaceutical Development Services at Excellience. He has a unique background in pharmaceutical development, combining extensive experience in contract development and pharmaceutical companies. At Excellience, his role is to provide leadership to the Pharmaceutical Development Services Group, which includes pre-formulation, analytical chemistry, and formulation development. Before joining Excellience, he held such positions as Vice President of Pharmaceutics at Quintiles, Director of Solids Formulation Development for Hecht Marion Roussel, and various management positions within Marion Merrill Dow and Marion Labs, which were predecessors to HMR. Dr. Skultety has a successful track record of developing new chemical entities from pre-IND to commercialization. He has 9 U.S. formulation and composition patents and has written a number of publications and made numerous scientific presentations. He has been a member of AAPS since its inception. And now, without further ado, I'd like to hand the microphone over to Paul Skultety. Paul, you may begin when ready.
Thank you, Donna. I would like to wish everybody a good morning or good afternoon, depending on where you're at. And thank you for joining us for today. Our discussion today is going to be on scale-up considerations, going from formulation development all the way through to commercialization. We're not going to have time today to really talk about the transfer aspect of dealing with this as well, but if you do a good job of what we talk about today during the scale-up process, the transfer process going from, let's say, the pilot plant into the commercial facility will be a very smooth and easy process. So today we're going to talk about what is really your formulation development objective, and we'll go through several phases from phase 1 to commercialization. We'll talk very briefly about some improvement approaches that we can utilize during the development process and even during commercialization. We're going to talk about understanding powders because that's very critical. That's if we can build a solid base when we're working with our powders from the very beginning, that again is going to make for a much smoother process when we get to large scale and when we get to commercialization. We'll talk about what do we need to consider during the scale-up, and then we'll do a very brief design of experiments film coating study, a case study that we've utilized. For our product development objective, clearly we want to develop a formulation and a process that will consistently deliver a dosage form with the desired quality attributes. And we want to create a robust formulation. And a robust formulation is a formulation that is able to accommodate the typical variability that's going to be seen in our API, the excipients we use, and the manufacturing process. And we want this variability to be handled so that it doesn't affect the stability or the performance of the product. We obviously want to be able to reproducibly manufacture the same thing time and time again. And our commercialization objective is we want the manufacturing process to be very well understood. We want our commercial manufacturer people to be able to have confidence that they're going to be able to manufacture this material over and over again, and that a minor change in the API or a minor change in any of the excipients would not cause them any over concern. So we basically want the variability to be managed by the process so we can handle a variety of changes in the raw materials. And the last thing we want to consider at the commercialization stage is we want to be able to facilitate continuous improvement. We want to be able to trend our data and show that everything is within specification and that we're not trending either one direction or another. So how do we want to be able to accomplish this? And we want to do it by being able to define our design space. And what do we mean by design space? And we'll talk about that in a little minute. But we basically want to propose our specifications for this product based on that evaluated space so that we know if we stay within this design space that time and time again we're going to be able to produce the same product. This will give us a lot more flexibility in proposing changes excuse me, with the FDA. If we have to adjust something or we want to change something a little bit because of changes in raw materials or changes in our equipment. As long as we stay within that design space that we studied, we shouldn't have to seek prior approval. We can do a change by effect submission to the FDA. And we want this design space, again, to account for the variability that we might see in either the raw materials, the excipients, the API, and the process. And we want to periodically review this because as we manufacture this over and over again, our knowledge base is going to grow, and we want to trend the data. And if there's any failures that we see, we want to take that into account. Unfortunately, if something goes well, we don't really learn a lot from that necessarily. But if something fails, we learn a lot from that. So unfortunately, in this case, from failure could come better results. And if you look at this picture, we basically want to evaluate a much wider space than what we're actually going to be operating in. So we call that our knowledge space, something that we've studied, but we don't necessarily want to routinely manufacture in there. Then we would have a tighter area from there that what we call the design space, that we know anything in that shaded area is going to provide the same product time and time again. And within that, which is normally where we're going to operate, is we will come into a tighter area as well. Now, to make this as best as possible, if we look at the knowledge space, we want it to be on the edge of failure. We want to know that if we happen to go outside of that area, that something might fail, and we will know why that might fail. That will help us in keeping everything well within our design space. There are three improvement approaches that we can utilize. One is what's called innovation, and we'll talk about this a little bit more. But this is a QBD-focused or quality by design-focused approach, meaning you want to build the quality in. You want to start from the very beginning when we first start with our excipient compatibility study and build the quality into the product all the way through to commercialization. and we want to evaluate and we want to optimize the process and formulation. And we want to do this by a statistical process. The second improvement approach is called continuous improvement. And this is really, we're manufacturing something over and over again, and we provide the product within specification each time we manufacture it. So we kind of do a little retrospective look at all the batches we've made. We collect the data and we evaluate the data, and we try to trend that data, and if need be, we might adjust that data. And last, which is not by any means the most preferred approach, would be a corrective action approach, where we look at the product deviations, we look at product characteristics that might not be as good as we want it to be, and then we try to do a risk assessment, excuse me, doing this approach, and a short-term fix, which would be reworking the material. Let's say we've manufactured a roller compaction and we end up with a lot of fines in that material on this particular lot. Well, we do a rework approach to send the fines back through the roller compactor. But again, this is not the preferred approach. We're not really building quality in at this point. We're trying to look for a corrective action for a problem that we're seeing. Now going back to the innovative approach, which again is our preferred approach, we really want to understand the process from beginning to end, from the weighing out of materials, the blending materials, all the way through to the finished product. And that will start with characterizing very well not only the API, but characterizing the excipients. Excipients can come in a wide variety of particle size, of different grades. So we want to make sure that we understand what is the effect of each excipient going to be on our material. And we want to do this evaluation based on very statistically valid experiments. It is very helpful if we have a statistician available that can help us, one, design the study so that we can get the most data out of the least number of runs, but also help us interpret the results when we get to the end. We want to make sure that we can define acceptable operating ranges, from the simplest thing to how long do we blend a material, to how hard can we compress it, what is the hardness acceptable range, to the coating bed temperature if we're doing coating, and so forth. And last but not least, we want to show that everything that we learned during this scale-up process, we can demonstrate at full scale. And we may need to do some challenge at full scale to show that the statistical design that we built up is valid at that level. So let's now just kind of step through the different phases that we go through. And what do we want to learn at each of these phases? When we're in phase one and we're doing a simple phase one study, we want to make sure that we're developing a prototype formulation. We want to try to start to lock in on what are the ratio of excipients and API and finalize that this is the group of excipients that we want. We want to start to identify the API physical characteristics and chemical characteristics to start to understand how is this material going to behave. We want to look at the particle size of the material and the flow properties, and we're also trying to hone in on what dissolution profile we're looking at. So basically, by the end of phase one, we pretty much want to have our prototype formulation selected. Going into phase two, and at the start of phase two, we're going to start up our scale-up process. And we need to scale it up to an appropriate scale. That is going to depend on what the processes we're looking at, what the drug is, how large we think the commercial batches are going to be. So there's a number of factors in there that will help you determine what scale you want to be at this point. And we want to start to look at the optimization of the process and the optimization of the formulation. We want to start to look at things like how much magstearate do we need to add to this formulation? How much Avicil do we need to add to the formulation? how long do we need to blend, or how long do we need to wet granulate, and how much water we might need to add for the wet granulation. So by the end of this, we want to be able to finalize our quantitative formulation for each of the excipients, and we want to start to hone in on what are the in-process and release specifications that we're going to need, and what are the important parameters for our formulation. So by the time we get to phase 3, we want to have a scaled up formulation for the pivotal batch, which needs to be within 10% of what our commercial scale is going to be, or a minimum of 100,000 dosage units. And you want that very close to the final formulation and process, so that when you go to file your NDA, there's not a lot of questions about what you put into your pivotal bio batch and what actually is going to be marketed. You can make some final adjustments to, let's say, mag stearate by processing it a little bit, but you don't want to be making large-scale changes at this point. And the other thing you want to keep in mind at this level is you want to make sure that all of your your analytical methods are validated. This will help you later on in making sure that everything is hunky-dory when you go to file the NDA. At the same time, we're going to manufacture our registration stability batches. And what do we need for those batches is we want to make sure that we have the final shape, the final color, any printing or embossing that will be on the tablet. Now the printing doesn't have to be the exact logo that's going to end up being on the tablet when it's commercialized. It can just say 1, 2, 3, it can say XYZ, same way with the embossing. It doesn't have to be the final embossing, it just should be on that tablet if you're going to have it for the commercial product. And as far as the packaging configurations, we need to know if we're going to be doing blisters, and if we're doing a blister, whether it's aluminum or a laminate in aluminum and so forth. And as far as bottles go, we need to know how small we need to go. Do we need to worry about doing physician samples of 5 to 10 in a bottle, or what is our lowest amount, and then what is the highest packaging configuration? The other thing we need to make sure we keep an eye on is the API synthesis should be finalized by now, and it should be very close to its final batch size. We don't want to see a lot of physical or chemical changes happening to the API at this stage of the game. The other thing we want to keep in mind about our rework procedure, or our transfer, or our scale-up, is rework procedures. We talked about roller compaction a little bit earlier. Again, if we know we're going to have fines and we want to reprocess those fines, we need to take that into account now at this stage. We also want to take into account any holding times for our materials. If we granulate a material, how long can we hold that before we compress it? Can it be a month? Can it be 2 months? That's something we want to look at. Or if we manufacture a coating solution, how long can that coating solution be held before we actually do the coating? And the goal at this point is that we're going to be ready for that NDA filing. And at the NDA filing and commercialization, we're really talking about everything's final. We've got the specification, the process, the finished products there. We've got our dissolution profile finalized, and we do now know what is the final print or embossing that's going to be on the tablets. We also have to take into account at this point validation batches. And if we do our scale up appropriately, the validation batches should go very smoothly. The question at this point is, normally companies would like to file the protocol for validation in their NDA, but they prefer not to execute those before the NDA is filed. The reason being is they would like to use those as their first 3 marketed batches. And because of this, they want to manufacture it as late as possible to keep as long of expiration date as possible. So our goal at this point is we want to pass that PAI when it comes up. We want to be ready for launch, and we want to be able to use SUPAC guidelines. If we have to make any changes, we can go by those guidelines. So let's now talk about the importance of powder and the characteristics of the powder, and why is it so important to us. First of all, the majority of APIs are going to be powder. So that's a key factor starting out there. And secondly, most manufacturing processes are going to involve powder. The majority of our dosage forms are powder. So that's a key thing that we need to keep track of. We want to be able to take into account the blending of the API and understand how the API and the excipients are going to blend. And we want to make sure that we get a good uniform blend, not only of the API, but also of the excipients. Particularly if we have some functional excipients in smaller quantities, such as a mag stearate or something like that. We also need to make sure that we understand, if we're doing a tablet, how it's going to flow and empty from the feed hopper, because a lot of times at a commercial scale, we're going to have hundreds of kilos in the hopper, not just a few kilos. And we need to make sure that the last few kilos coming out of that hopper is the same quantitative formulation as the first few kilos coming out of that hopper. We also need to make sure that it fills in the feed frame and the die cavity, and that the mag stearate has been very well dispersed so that we don't have any problems on compression or the ejection of the tablets. So the flow properties of this powder becomes very critical. So we have to understand how not only do the individual things flow, but how well they flow together. So we're going to look at such things as bulk and tap density, and we'll talk a little bit later about that as well. We want to look at the shear properties of the material and the compressibility of it. We want to look at the particle size and shape and distribution of our material. So particularly the API, if it's in a high concentration in the formulation, and if that particle size changes any, we want to know what the effects are going to be on the formulation, and whether or not we're going to have any concerns as far as solubility goes if something changes in our material. Or what about the moisture content? If we're doing a wet granulation, what does that moisture do to this material? And if we dry it too much, are we going to have a problem with electrostatic charge? And as far as the shape goes, this just shows you to an extreme what you can possibly get as far as a plate-like material for either one of your excipients, or in this case a particular API. And this isn't too bad if you know up front and you deal with it, but if all of a sudden you have a material that starts to have these plates that continue to get larger and larger, the flowability of this material is going to continue to become more of a problem. And the other thing we want to take into consideration is if we start to see large changes in our particle size of any material, you're going to start to see a very dramatic change in the surface area. And where this can become a concern is if we're doing a mag stearate that we're using to coat material to prevent sticking, if all of a sudden our surface area becomes much larger, the amount of mag stearate that we have in the formulation might not be sufficient anymore to do appropriate lubrication. So the next thing we want to talk about is the CARS index. And we want to be able to use this as an indication of how our material is going to behave. A CARS index is determined by taking the tap density minus the bulk density divided by the tap density times 100. In our sample calculation here, we come up with a CARS index of 28%. It was fairly common tap densities of about 0.6 and a bulk density of about 0.4. And what we can use this for is that we can estimate how our powder is going to flow and what is it going to look like. And these are by no means etched in stone that a material that has a CAR index of, let's say, 25 is going to be a poor flowing versus a CAR of 24 that's going to be fair. These are just kind of rough guidelines that you can use to say, okay, if you know your material is going to have a CAR index of somewhere above 18, flowability may ultimately become a problem. If you have a car index of something less than 17, probably flowability is not going to be too much of a concern for what you're doing. If we talk about the API, and we know we need to make sure that we take that in consideration for our scale-up, what properties do we need to make sure that they're considering when they're doing their scale-up? And we need to make sure that the chemical stability doesn't change, sensitivity to moisture or light, which usually is not a problem. Solubility shouldn't change too much, but where we can sometimes see a problem is all of a sudden we start to see some polymorph issues, where what used to be a fairly pure material now all of a sudden starts to see a little bit more of a polymorphic change. The other thing that is much more common of a problem is the particle size, shape, and or particle size distribution curve dramatically changes. And that's where we can start to see problems when we're doing our work as far as scaling up. So we need to make sure that whatever material we're going to use for our scale-up is going to be very representative of the material that's going to always be supplied. Because otherwise we could spend a lot of time and effort doing our DOE work when in reality we're using the wrong material for that. And some of the critical factors, if we look at what type of dose that we're looking at for the active, what do you need to be concerned with? If it's a high dose material such as greater than 250 milligrams or somewhere around that, again, these are not hard and fast rules, it's just a rough guideline. We need to definitely be very concerned about the flowability of the API. We also need to be concerned about the compressibility of the API and the availability of the API, and we'll talk about that a little bit more in a minute. We want to look at mid-range dose of somewhere around the, let's say, 25 to 250 milligrams. And availability may not be as much of a problem depending on where you're at in that range, but definitely flowability is still a problem, or a potential problem, I should say, not necessarily a problem. And if we're looking at the low dose, things that are less than 25 milligrams, handling procedures might be a concern if this is a very potent compound. Stability might become a concern, but much more so as content uniformity becomes more of a concern. Because the dose is much less, flowability usually isn't a problem unless you're trying to make a very, very small tablet. Something around, let's say, a 75 milligram tablet or so, total tablet weight. But usually when you get to the lower doses, you can dilute out the concerns about flowability of the API. And this is something that I like to bring up because it's something that people a lot of times will forget, is for all of our design of experiments and everything that we do, we need to make sure that we have the appropriate amount of API available. Because once we start dealing in larger batches and we do a number of experimental batches that you need to do for your DOE work, you consume a lot of API very quickly. And just looking at this, even for a batch size as small as 50,000 tablets, which is not a very big batch, if your strength is around 100 milligrams, you're using up 5 kilos at a batch. And if it's 500 milligrams, you're using up 25 kilos at a batch. So you're consuming a very large amount of material very quickly. And if you don't plan this into your API manufacture schedule, you're not going to end up with the appropriate amount of material at the time that you need it. So if we look at how getting more into the scale-up concern, the design of experiments, and what are our objectives? Like we mentioned, we want to determine that design space. We want to make sure that we're providing the optimum manufacturing parameters that can provide something that's very reproducible. We want to minimize any batch-to-batch variation and support scientifically the in-process, and the release specifications. I want to take the time here to define two terms. A critical process parameter, which is what we want to determine is which of our parameters are critical. And this is something where the variability could impact a critical quality attribute of our product. And a critical quality attribute of the product is something that must be controlled to ensure that the product meets its intended safety, efficacy, stability, and performance. So when should we start the DOE work, and when do we really start considering our scale-up? We want to make sure that we have an established process and that the equipment train is determined so that we know we're going to do, let's say, a blend, a roller compaction, a blend with a lubricant and compress, or we're going to do just a direct blend and compress, or we're going to do a wet granulation, or whatever it might be. But we need to make sure that we have that train determined before we start doing a lot of scale-up work. Otherwise, it could all change, and that work is for naught. We usually want to start this no later than phase 2B and definitely prior to phase 3. And we also need to know what our strengths are going to be for that particular active, or at least a range that we're going to be working with. We need to know, are we going to be working with a 25 milligram tablet or a 250 milligram tablet? And why this can become so difficult is, let's just take a look at a process of a simple wet granulation, compression, and coating for a tablet. And these are the different process factors that you need to evaluate to see which of these might be a critical parameter that you need to study. So we need to determine which of these are a critical process parameter and which of these might affect the critical quality attributes. Now, hopefully we don't have to study each of these, but we do need to make sure that we address each of these. And the responses that we would look at are typically such things as this. For the powder blend, we want to look at the content uniformity. For granulation, we want to look at the size and shape and distribution for the particles. We want to look at the density, both bulk and tap density. We want to look at the flow rate, moisture content, and again, content uniformity. And for tablets, we'll look at such things as weight and hardness and so forth. So the ideal experimental design is going to be a screening study first so that we can determine which of those process parameters might be a critical process parameter. And we'll look at something like a Plackett-Berman design where we can do anywhere from 8 to 12 experimental batches at an appropriate scale. We want to review historical data and help us if we can eliminate some of the variables. We want to study as many parameters as we can and that's the reason we would do a Plackett-Berman screening design. We want to identify, like I mentioned, what are the critical parameters. And then we also want to perform, before we start this study, we want to perform some trial runs at the extreme conditions so that we can make sure that we can manufacture and get data from each of the runs that are going to be in this Plackett-Berman design. And what I mean by that is that we want to make sure that if we're doing a wet granulation and we're varying the amount of water being added, that at some point we don't add too little water, that we don't granulate at all, or that at some point we add too much water to it and that we end up with just a ball of material that we can't do anything with. So we want to make sure that any time that we're doing this, that we can do the work for all the runs. Otherwise, if we lose one of the runs when we do the study, that will really throw off your statistics. And then once we've done the Plackett-Berman, we would really like to follow that up with a full factorial of some point with center points. And the reason we do this is we want to characterize the magnitude of the effect. We use the center points to estimate what our variability is in the process. And that will help us, again, get the best information out of it as we can. We want to try to establish if there's any interactions between any of the parameters. And then we want to get to the point that we can do a response surface. So that can really help us define our design space. This will give us scientific justification for the manufacturing parameters. And we will usually end up doing multiple studies. We will do blending study, and then we'll identify what is the best blend parameters and process. We will take that process and do the granulation study. And then once we determine that, we will take the best of both of those and then do the compression study. So we don't do a blend all the way through. We optimize each step along the way. And again, we have to take into account and make sure that we can perform the trial at each of the variables that's in there. So let's now talk about a case study for some bead coating, and just to show you real quickly what type of data you can do with the properly designed studies. So our process parameters that we looked at for this is we were looking at spray rate, atomization pressure, product temperature, humidity, and pan speed. Now the first thing we looked at was we looked at the particle size that we got, both small scale and full scale, to make sure that our particles are going to be roughly the same from beginning to end. We ran our study, and we were looking at two different time points for our dissolution. We were looking at 30 minutes and 60 minutes. And this is a statistical analysis of the different parameters that we looked at. We found one that was significant interaction between, one was significant. The interaction between spray rate and temperature, and the interaction between humidity and temperature was not significant. In this particular table, any value that is greater than 5 was considered to be significant. Someone has asked a question, in my experience, what are the most typical issues and challenges that innovators face during scale-up? Probably, I think the biggest challenge that I see is when you start to have variability in your raw materials or your excipients and the API, when the API is constantly changing, so that it's very difficult to build up a database. The second biggest challenge I see is lack of API, so it becomes very difficult to scale the material up, meaning there's no drug available to do a lot of work at a larger scale. Part of it depends on the process. If you're doing a direct blend and tableting, that's much easier to scale up than if you're doing a wet granulation. Part of that depends on issues would be, do you have the same equipment train going from beginning to end? Meaning, if you're doing a wet granulation on a small scale, does it operate the same as what you would see on a larger scale? And that's probably where I see more problems than anything else, is just trying to keep consistency of both your raw materials and equipment, and having enough API available to be able to do the work appropriately. A lot of times you're asked to do things very quickly with not enough material. You've been working at a scale of, let's say, 500 grams, and then all of a sudden you're asked to jump up to 25 kilos or more. And depending on the process, that can be a very big jump, and it can be sometimes very challenging to come up and make sure you get the same material. So going back to our presentation here, what we can do with this data that we collect is, as I mentioned, we can put together these type of plots, and you can look at what is the effect of these different variables, pressure and temperature, on the dissolution amount that you get at 3 hours. And the other thing it shows you is that at certain conditions it may have a positive effect, and at certain conditions it may have a negative effect. So it's very important to look at this because as you see in this particular case, if you look at your amount dissolved at 3 hours, it varies from about 2% all the way up to 40%. So you get a very wide swing in this particular amount. And in this particular case, if we look at the effects of pressure and temperature, what you can see is almost a saddle effect where you're going from a high at 44, you then go down through a minimum, but then you end up back at almost the same amount again as well. So it's not as easy as this is going to be a linear plane that goes through, but you can also have saddle points as well that go through here. So you have to take all of these into consideration. And in this particular case, if we're looking at these conditions and looking at the effect of humidity and spray rate on pressure and temperature, excuse me, you see very minimal effect. You can vary your temperature from 24 up to 30, and you can vary your atomization pressure from 2 up to 4, and you're getting very, very minimal effect on your dissolution. It's basically going from 19% up to about 21%. So in some cases, your variables will have a large effect on your result, and in some cases, such as this, your variables are going to have very minimal result. So basically, we've tried to very briefly step through today what is your objectives during the scale-up process. It's very hard to define a particular scale or anything like that because a lot of it's going to depend on what the project train is going to be. It's going to depend on how much API is available. It's going to depend on what your critical processing parameters that you identify are. But basically, I think the message that we want to take away from this today is that the more you can study this early on, the easier it is going to be to transfer it to a commercial facility and to manufacture it reproducibly commercially. But the key is that you want to be able to at least do some minimal work, design of experiments or however you want to call it, to help you identify what is the design space that you can operate in to still reproducibly manufacture that product. And if you set this up appropriately and you follow the steps and go through doing the DOE type of work and doing that type of statistically designed process, then what you'll end up with is you'll have a product that is quality by design. And if you get to that point, then it's going to be much easier to get your PAI through with very, very minimal questions at all. So now at this point, I've concluded my presentation. If there are any questions that you would like to ask, I would be more than happy to try to answer them.
Thank you very much, Paul. Yes, we're now into the Q&A part of our program. So at this point, I would like to invite you to continue to send in a question or comment using the panel on the right-hand side of your screen. And here we have a question for you, Paul. How are regulatory requirements driving this process? And can you talk a little bit more about quality by design?
I can talk a little bit about the quality by design. And quality by design is a very big umbrella that really is nothing more than what a lot of people in the industry have been doing for many, many years. It's basically you're trying to scientifically be able to, as we mentioned, determine your design space and be able to scientifically say, these are my operating parameters and this is why you want to operate it in that range. And it can go, quality by design really starts when you're doing excipient compatibility work all the way through to your scale up into commercialization. Excipient compatibility is really part of quality by design. You're starting at the very beginning and saying, these excipients are going to be compatible with my drug. And this is why. And we've done the testing to show that. And then you really build upon that as you go through the development process. And the other part of that question was how are regulatory requirements driving this process? Basically, everything we do is really driven by regulatory requirements. That's really what GMP is all about. But beyond the regulatory requirements, when you're dealing with something that you're going to commercialize and that you want to be able to make 10 batches a year, 20 batches a year, however many batches a year that you need, then you really want to build as much science into that as possible. And the other part of that is with having the SUPAC guidelines, we can make changes in the commercial process and formulation if need be without having to go and get prior approval. You can do a lot of things now with CBE or change by effect, and you can just file that to your NDA, and as I mentioned, then you don't have to get prior approval for that type of change. So that's really the big advantage of doing a lot of this and doing it by so-called quality by design, is then you can use things to your advantage when you get into commercialization, such as the SUPAC guidelines.
That's good. Thank you very much. And that is all the time we have for today. I'd like to thank everyone for attending today's webinar. And don't worry if you missed part of Paul's presentation. Simply visit www.excellience.com, where you can download any of our archived scientific seminars. If you want to share this presentation with your colleagues, simply note their name and email address in the post-webinar survey. We will then send them a link to the archived presentation. Please join me in thanking our speaker for today, Dr. Paul Scultety, who is Director of Pharmaceutical Development Services at Excellience. You will notice that a post-event survey will pop up on your screen at the end of today's seminar. Please fill out this survey because we would really appreciate your feedback as well as your suggestions for future webinar topics. Thank you all for your time. We hope you found this webinar informative. Have a great day.